ultra-slow oscillations in fMRI and resting state connectivity, neuronal and vascular contributions and technical confound. Patrick J. Drew, 1, 2, 3. Celine Mateo, 4. Kevin L. Turner, 2. Sin Yu, 5, 6, and David Kleinfeld, 4, 7, 8. Abstract. Ultra-slow, 0.1 Hz variations in oxygenation level of brain blood is widely used as a fMRI-based surrogate of resting state neuronal activity. The temporal correlations among these fluctuations across the brain are interpreted as functional connections for maps in neurological diagnostics. Ultra-slow variations in oxygenation follow a cascade. First, they closely track changes in arteriole diameter. Second, interpretable functional connections arise when the ultra-slow changes in amplitude of gamma-band neuronal oscillations, which are shared across even far-flung but synaptically connected brain regions, entrain the 0.1 Hz vasomotor oscillation in diameter of local arterioles. Significant confounds to estimates of functional connectivity arise from residual vasomotor activity as well as arteriole dynamics driven by self-generated movements and subcortical common modulatory inputs. Lastly, methodological limitations of fMRI can lead to spurious functional connections. The neuronal generator of ultra-slow variations in gamma-band amplitude, including that associated with self-generated movements, remains an open issue. Go to Introduction. How do brains compute? The answer to this question takes on many guises. It has proven useful to differentiate brain regions on the mesoscopic scale, i.e., as an average over large number of individual neurons, and thus define connections and signaling on the same mesoscopic scale. Mitra, 2014. Zhang, 2018. As an example, the flow of sensory input in the transformation from perception to action can be described as a flow from primary sensory areas to motor areas. Ferezu et al., 2007, Li et al., 2016. Mesoscopic connectivity diagrams are readily determined based on the histological analysis of tissue subsequent to the injection and transport of anterograde and/or retrograde markers. Boland et al., 2009, Kim et al., 2017. However, the determination of connectivity within the living animal and the modulation of signaling along established axonal pathways as a function of brain state requires a non-invasive procedure. A well-established, albeit incompletely understood approach is based on functional magnetic resonance imaging, fMRI, an imaging modality that infers neuronal activity from changes in localized brain blood oxygenation. Logophetis and Wandel, 2004, Schulman et al., 2002. Here we review the use of fMRI to infer neuronal connectivity between well-separated regions in the forebrain. These are correlation-based methods, and regions with strong correlations between their resting state signals are said to be functionally connected. Connected graphs that are comprised of regions as vertices and strong correlations as edges are said to form default networks of brain activity. Gressius et al., 2003, Reichler et al., 2001, Sporns et al., 2005. Such networks have been characterized in humans. De Luca et al., 2006, De Luca et al., 2005, Majid et al., 2011, Zhao et al., 2008, monkeys, Shemuel and Leopold, 2008, Vincent et al., 2007, and rats, Ghazi and Schwartz, 2016, Liu et al., 2012, Majid et al., 2011, Stafford et al., 2014, Thompson et al., 2013, albeit not without controversy as to reliability, Honey et al., 2009. Our review generally avoids the discussion of signals from anesthesia animals and humans in light of the strong disruption in the interaction between neurons and blood vessels by most anesthetic agents. Gao et al., 2017, Gintz and Logophetis, 2008, Pissarro et al., 2013. Go to Fundamentals. Both the flow and oxygenation of blood in the brain is modulated by neuronal activity. This modulation forms the basis of fMRI, an intrinsic optical signal (IOS) imaging. Two coupled effects are at work. First, an increase in neuronal activity will lead to an increase in the diameter of the pile arterioles that distribute blood to the brain. This change in diameter will long outlast the stimulus. Drew et al. 2011, Ulirova et al. 2016, Figure 1a. Second, an increase in neuronal activity will initially lead to a decrease in blood oxygenation in the microvessels and veins, seen most prominently in the upper layers of cortex. Buxton, 2001, Grinville et al., 1991, Tian et al., 2010. Albeit this interpretation is not universally accepted. Sinkin et al., 2020, Saratin et al., 2009, Ulyadeg et al., 2009. The increase in oxygen consumption is soon overcompensated by an increase in blood flow and a concomitant increase in blood oxygenation. The overcompensation reflects the active nature of neurovascular coupling. Atwell et al., 2010, Colley and Hamill, 2010, Iodicola, 2004, Kleinfeld et al., 2011. The overcompensation can localize to one or few cortical columns. Grinville et al., 1991. Yet it may have a global component from dilation of arterioles caused by change in the activity of sympathetic input. Dacey Jr. and Juling, 1984. An external file that holds a picture, illustration, etc. Object name is NIMES 1634830F001.jpg. Figure 1. The neocortical vascular response to phasic sensory stimulation and the mechanism for fast neurovascular coupling. A. The canonical effect of neuronal activity on the diameter of pile vessels as measured with two-photon laser scanning microscopy, Kleinfeld et al., 1998, using a head-fixed rat. The stimulus is a 500 milliseconds puff to the vibrissae. Adapted ROM, Ularova et al., 2016. B. The canonical effect of neuronal activity on hemodynamics as measured with intrinsic optical imaging using 630 nanometers light, Grinville et al., 1988, using a head-fixed mouse. The stimulus is a force puff to the vibrissae. The isoflurane data refers to a breathing mixture of 1.8% VV, isoflurane and oxygen, adapted from Knudsen et al., 2016. C. Rapid mechanisms for communication of neuronal activity to drive smooth muscle hyperpolarization and arteriole dilation. Neural activity 1 leads to an increase in K+ in the perivascular space surrounding microvessels that activates the potassium inward rectifier, 2, KIR, to generate a local hyperpolarization of the endothelial membrane. 3. The hyperpolarization spreads to adjacent endothelial cells through gap junctions to activate KIR currents in adjacent cells. This results in a propagating signal from microvessels to penetration arterioles and finally surface or pile vessels. The hyperpolarization spreads to adjacent smooth muscle cells, 5, and deactivates voltage-dependent Ca2+ currents, CAV, to induce smooth muscle relaxation and arteriolar dilation. 6. Rapid arteriole dilation is also induced by binding of nitric oxide, Neuronal, that is released from some inhibitory neurons, as well as other brain cells, i.e., endothelial cells, astrocytes, and microglia. The increase in arteriole diameter, panel A, can lead to an increase in blood flow. Adapted from Longden et al., 2017, and Knott and Nelson, 1998. The oxygenation state of blood can be detected via differences in the electronic properties of oxy versus deoxyhemoglobin. This is reported by a greater reflection of red light by oxy versus deoxyhemoglobin, which is routinely detected by IOS imaging, Leak et al., 1989, Figure 1b. It is also reported by a change from a diamagnetic to a paramagnetic state by oxy versus deoxyhemoglobin respectively, Pauling and Coriell, 1936. Thus, the magnetic susceptibility of capillary and venous blood is correlated with neuronal activity. The change in arteriole diameter and blood oxygenation are coupled. The overcompensation of blood oxygenation is quenched when the pile arterioles are fully dilated using isoflurane prior to the onset of neuronal activity, Figure 1b. Knudsen et al., 2016. Communication from neurons to arteriole smooth muscle. How do neurons communicate with arterioles? There are multiple mechanisms that neurons in the brain use to control their own supply of blood. Atwell et al., 2010, Colley and Hamill, 2010, Iodicola, 2004, Kleinfeld et al., 2011. We review two mechanisms that acts on a less than 1s timescale. The lumen of the blood vessels is formed by endothelial cells that are connected with relatively low resistance gap junctions, such that passive signaling occurs within 2 mm electrotonic length. Siegel and Juling, 1989, Welsh et al., 2018. Further, the smooth muscle cells that drive vascular tone are in strong electrical contact with the endothelial cells. In the presence of bursts of neuronal spikes, there is an outflow of K+ ions in the perivascular space. Caesar et al., 1999, Rasmussen et al., 2019. This can lead to regenerative, hyperpolarizing pulses that propagate along the endothelial cells from the subsurface microvessels to the penetrating arterioles, Longden et al., 2017, and then to pile arterioles, Figure 1c. The hyperpolarization will spread to smooth muscle and dilate the pile arterioles, Filosa et al., 2006, Knott and Nelson, 1998. Insert in Figure 1c. This sequence of activation is supported by the stimulus-driven progression in arteriole dilation from middle layers to the pia and cortex, Tian et al., 2010. While details of the conditions for passive versus active spread of signals in the pile network remain to be addressed, Harris et al., 2018, direct electrical coupling through the endothelium provides a means for neurons to directly influence the diameter of neighboring arterioles, Figure 1a. Nitric oxide NO is a well-known vasodilator that is produced by nitric oxide synthase NOS in neurons and can act quickly to relax smooth muscle, Figure 6. As a caveat, the role of NO as a transient actuator of change in arteriole dilation, et al., 1996, as opposed to a cofactor in affecting change, Lindauer et al., 1999, remains unsettled, Atwell et al., 2010, though optogenetic stimulation of neurons that express no synthase will drive an increase in blood flow, Krawczyk et al., 2019. The role of astrocytes in the control of flow is also controversial, Kleinfeld et al., 2011, Mishra et al., 2016, Nizar et al., 2013, Rosenegger et al., 2015, with arteriole dilation occurring only in the presence of relative large signaling events by astrocytes, Schultz et al., 2012. An external file that holds a picture, illustration, etc. Object name is NIMES 1634830F006.jpg. Figure 6. The envelope of neuronal gamma band oscillations locks to and leads vasomotor oscillations in arteriole diameter. A. Setup with a head fixed awake but resting mouse for 2-photon imaging of arteriole diameter along with measurement of the LFP, left panel. A thin skull window was used to preserve vasomotion, Drew et al., 2010. 2-photon images of surface vessels and scan path to define lumen diameter, center panel, from Mateo et al., 2017. B. Example data showing the LFP, top trace, the spectrogram of the LFP with a window of 2.0 s and a bandwidth of 2.5 Hz, and the time series of the integrated gamma band power and diameter for one arteriole in the field, bottom traces, from Mateo et al., 2017. C. The cross-correlation of the two time series used for the example of panel B, 600s total time, show that the LFP leads the diameter change. As an average across animals, movement precedes the vasodilation by 1.9 plus or minus 0.2 s, 82 vessels from 27 mice with 600 s of data per vessel. From Mateo et al., 2017. D. New data on the recording of self-generated, i.e. spontaneous movement in a head fixed awake but resting mouse, obtained concurrent with 2-photon imaging of arteriole diameter, Winder et al., 2017. Similar to that in panel A, to determine the possible contribution of such movement to the entrainment of vasomotion. Bouts of whisking were determined with videography, and whole-body vertical acceleration determined with a force sensor attached to the tube supporting the mouse's body. E. Correlation analysis of new data, 27 vessels from 9 mice with 1,700 to 12,000 s of data per vessel. The time lag of the correlation shows that movement leads the change in diameter, bottom panel, with lag time similar to that for movement precedes the vasodilation by 1.8 plus or minus 0.3 s for whisking and 1.5 plus or minus 0.3 s for whole body motion. F. A reanalysis of the data of Mateo et al. 2017, similar to that for the new data of panels E and F, using previously unpublished simultaneous measurements of the whole body horizontal acceleration acquired during imaging, 82 vessels from 27 mice. Accelerations lead to a change in arteriole diameters. Left panel, the threshold detectability was 0.01 grams. Theses movement-related changes in diameter are compared to rest events, middle panel, and dominate the largest changes in diameter, right panel. Background on fMRI. The blood oxygenation level dependent (BOLD) fMRI signal exploits the shift in the magnetic properties of deoxyhemoglobin versus oxyhemoglobin, Villringer and Dernagel, 1995. In practice, these are detected by changes to the relaxation time of water protons in the brain, Ogawa et al., 1993, Stefan et al., 2004. Longer relaxation times implies greater oxygenation and a larger bold fMRI signal. The bold effect was discovered in 1990 by Seiji Ogawa using anesthetized rats, Ogawa et al., 1990. 2 years later, bold was independently demonstrated in the awake human brain by the laboratories of James Hyde, Bondatini et al., 1992, Bruce Rosen, Kwong et al., 1992, and Kamil Ugerbil, Ogawa et al., 1992. As an example, successive eye opening and closing in humans leads to a change in the bold fMRI signal in the brain region that corresponds to primary visual cortex, Fox and Reichler, 2007, Figure 2a. In terms of impact, the use of bold fMRI has stormed across the disciplines of psychology and cognitive neuroscience as a means to infer changes in neuronal activity during all aspects of cognitive tasks performed by humans. An external file that holds a picture, illustration, etc. Object name is NIMES1634830F002.jpg. Figure 2. Functional MRI, fMRI, uses correlation in the fluctuations of signal strength to infer brain regions that are directly connected. A. Changes in the basic blood oxygenation level dependent (BOLD) fMRI signals generated in human visual cortex in response to changes in luminance in a full-field visual input. Unaveraged bold signal, magenta, was obtained from a region in the primary visual cortex during a task that required subjects to open and close their eyes. The task is shown in blue, and the times are delayed to account for the hemodynamic response. The subtraction of the eyes closed condition from the eyes open condition identifies a bold signal intensity difference in the primary visual cortex, shown on the right. Note the variability as well as the average increase in signal amplitude upon stimulation, from Fox and Reichler, 2007. B. Blood oxygenation level dependent fMRI signals were obtained from a coronal slice, 2.8 centimeters from the occipital pole left. 3 unprocessed time courses of the bold fMRI signal of representative voxels, numbers 1 to 3 and located outside of visual cortex, and their corresponding frequency spectra, right. A binocular visual stimulus, provided by a pair of flickering red LED patterns, was on during the 40 to 70th time interval of the 110th period and drives voxel 4, data not reproduced here, from Mitra et al., 1997. C. The bold fMRI signals recorded from 2 distant regions in human cortex, the posterior cingulate precuneus cortex (PCC) and the medial prefrontal cortex (MPFC), are highly correlated on the timescale of tens to hundreds of seconds. Correlations are calculated between a seed voxel in the PCC and all other voxels in the brain for a single subject during rest. The spatial distribution of correlation coefficients shows both positive and negative values and was thresholded at 2 South African Rand equals 0.1, left image. The time course for a single run is shown for the seed region, PCC, and a region, MPFC, positively correlated with this seed region. The functional connection between the PCC and the MPFC is consistent with a fiber track revealed by diffusion tensor imaging, blue trace in right image. Left and center panels from Fox et al., 2005, right panel from Grecius et al., 2009. D. 4 well-matched pairs of resting state subnetworks that show a close correspondence between the independent analyses of activated, left column, and resting state, right columns, brain dynamics. Activation data is from the 20-component analysis of the 29,671 subject brain map activation database, Fox and Lancaster, 2002, Laird et al., 2005, and resting state data is from a separate analysis of a 36-subject resting state fMRI dataset, Smith et al., 2009. Shown are the three most informative orthogonal slices for each pair. Left columns, network from brain map, shown superimposed on the MNI 152 standard space template image. Right columns, resting state fMRI data, shown superimposed on the mean fMRI image from all subjects, from Smith et al., 2009. A complementary technique, cerebral blood volume, CBV, fMRI, infers neuronal activity from an increase in total hemoglobin in response to neuronal activity. It was first demonstrated in the human brain using subjects whose blood was doped with gadolinium, a paramagnetic ion that vastly increases the magnetic susceptibility of blood, Beliveau et al., 1991, Kwong et al., 1992. The increase in blood volume is secondary to an increase in arteriole diameter, Figure 1b. Subsequent work has demonstrated CBV fMRI using label-free procedures, Huber et al., 2017, Liu et al., 2003. Noise and the determination of correlated brain activity. The physicist Louis Branscombe famously wrote, "God loves the noise as much as the signal." Branscombe, 1995. The noise in the fMRI signal, a part of physiological contributions such as breathing and technical noise, has dominant albeit broad spectral bands, Mitra and Bokil, 2008, Papoulis, 1962, near 0.1 Hz, Figure 2b. The exact frequency can vary on the scale of centimeters across the human brain. Mindful of Branscombe's prescient comment, only a few years after the advent of bold fMRI, additional experiments in the laboratory of James Hyde showed that the variability observed in the bold fMRI signal is correlated across well-separated regions in the brain. The initial observations involve subregions in motor cortex of the two hemispheres whose bold fMRI signals co-fluctuate in the vicinity of 0.1 Hz in the absence of a task, Biswal et al., 1995. These signals, in contrast to stimulus in task-based fMRI, are measured in the absence of a cognitive task or any overt stimulus and are referred to as resting state fMRI signals, Biswal et al., 2010. They are thus referred to as the resting state, bold fMRI. In fact, the signal between any pair of well-separated regions in the brain will co-fluctuate so long as they maintain long range, i.e., white matter, connections, Fox et al., 2005. This is illustrated for resting state bold fMRI signals from the posterior cingulate cortex, PCC, and medial prefrontal cortex, MPFC, left and center, Figure 2c, which are connected by a white matter tract along the cingulum bundle, Gressius et al., 2009, right, Figure 2c. In general, resting state fMRI signals are particularly strong between regions in opposing hemispheres with mirrored neuronal activation, Smith et al., 2009, Figure 2d, albeit these signals are accompanied by brain-wide activation, Skalvink et al., 2015. Arteriole diameter naturally oscillates near 0.1 Hz. What are physiological attributes of blood vessels that allow them to be coupled to ongoing neuronal activity in the absence of a task, Figure 2c, d, as opposed to be driven by strong sensory stimuli, Figure 1? Further, what is the origin of the 0.1 Hz frequency scale for the ultra-slow dynamics that dominate the resting state, bold fMRI signal, Figure 2b? Both of these questions are answered by recalling that arteries undergo vasomotor oscillations as part of their normal physiology, Intaglietta, 1990. These are collective oscillations of contractile tone in the smooth muscle cells that surround arterioles and lead to rhythmic changes in arteriole diameter, Alkger et al., 2011, Intaglietta, 1990, that are referred to as vasomotion. The rhythm thought to be generated by the interactions of active conductances in the membranes of smooth muscle and endothelial cells, Alkar and Nielsen, 2005, Haddock and Hill, 2005, with the mechanical forces generated by flow, Königsberger et al., 2006, Sturgiopoulos et al., 1998. Vasomotion occurs within a broad frequency band that is centered near 0.1 Hz in humans, Nordmans et al., 2018, Obrig et al. 2000, Reshupsky et al. 2013, Mice, Drew et al. 2011, and Rats, Kleinfeld et al. 1998, Mayhew et al. 1996. Vasomotor oscillations in arteriole diameter drive oscillations in the velocity of red blood cells in the microvessels, Drew et al. 2010, Kleinfeld et al. 1998, and changes in the partial pressure of oxygen in brain tissue, Mateo et al. 2017, Mayhew et al. 1996. As an intrinsic property, vasomotor oscillations are observed in isolated brain arterioles that are cannulated, pressurized, and maintained at physiological temperature, Assault and Halpern, 1988, Figure 3a. Critically, vasomotion is observed in pile arterioles in vivo, left, Figure 3b, and is only slightly reduced in amplitude, by a factor of 0.2, when cortical activity is silenced to isolate the vessels from ongoing cortical, albeit not sympathetic, neuronal activity. Winder et al., 2017, center and right, Figure 3b. An external file that holds a picture, illustration, etc. Object name is NIMES1634830F003.jpg. Figure 3. In vitro and in vivo measurements of vasomotion, a rhythmic change in arteriole diameter with a broadband intrinsic frequency centered near 0.1 Hz. A. Simultaneous recording of lumen diameter, top, and temperature, bottom, in a cerebral artery that was isolated from a spontaneously hypertensive, stroke-prone rat and pressurized to 80 millimeters of mercury. Diameter oscillations appear when the artery is warmed above 35 degrees Celsius, from Assault and Halpern, 1988. B. Example of normalized diameter measurements from a single pile arteriole with 2-photon laser scanning microscopy following local artificial cerebral spinal fluid, AXF infusion, left, and muscimol infusion, center infusions in a single animal. The population summary of the root mean square deviation in pile arteriole diameter after muscimol infusion, 19 vessels from 4 animals. Oscillations amplitudes were reduced following infusion of muscimol compared to AXF, from Winder et al., 2017. C. 2-photon imaging of vasomotion in cortical arterioles across both hemispheres of an awake, head-fixed mouse. A maximally projected image stack across 500 m of the preparation was obtained with ultra-large field of view, 2-photon microscopy, Tsai et al., 2015, top row. The dark region in the middle corresponds to a physical mask. The arbitrary line scan path of the beam, yellow, spans both hemispheres. The expanded images, middle row, are single planes in each hemisphere and serve to highlight the path of the line scan through individual vessels whose diameters were concurrently monitored. Vasomotor oscillations measured simultaneously from pile arteries in the right, green, and left, red hemispheres appear synchronous, middle panel in middle layer. The full dataset of 600S is seen to be correlated with a lag time at the peak of 0.0 plus or minus 0.1 s, lower row left panel. The spectral coherence in the diameter from pairs of arterioles, 24 traces of 540 traces, calculated with a bandwidth of 0.04 Hz as an average over 24 measurements, from Tsai et al., 2015. The presence of ultra-slow natural oscillations in the diameter of brain arterioles raises 3 questions. First, what is the relation of the oscillation to the change in oxygenation of the neighboring tissue, i.e. the parenchymal contribution to the bold signal? Simultaneous measurements of the arteriole diameter and the intrinsic optical signal in awake mice show that the two are strongly coherent, Mateo et al., 2017. The peak value of oxygenation lags the peak of vasodilation by 1s. The positive coupling of blood oxygenation with a change in arteriole diameter is consistent with the changes that occur upon sensory stimulation, Figure 1. The second question, given the coupling of blood oxygenation to vasomotion, concerns the spatial scale of synchronous vasomotor oscillations. The results from direct measurements of vessel diameter in awake rodents indicate that vessels within a radius of 2 mm tend, on average, to dilate synchronously, Mateo et al., 2017. Further, since the radius is the only dimension of blood vessels that can change, CBV fMRI provides a measure of arteriole diameter. Using this technique with anesthetized rodents, A correlation length of 2 mm is also reported, he et al. 2018. Thus, despite the short-range falloff, are changes in diameter strongly coordinated across distant but mirrored regions in the two hemispheres, Figure 3c? Optical imaging data from rodents shows that such transhemispheric changes are strongly correlated, Mateo et al. 2017, Tsai et al. 2015. Similar results are inferred with CBV fMRI of the primate brain, Skalvink et al. 2010. All told, mirrored regions in the two hemispheres show synchronous oscillations in arteriole diameter, Figure 3c, in addition to blood oxygenation, Figure 2d. The third question is how fast the callosal coupling between neurons in opposite hemispheres must be to support synchronous ultra-slow activity. For the simplest models of weakly coupled oscillators, this means a delay of less than a quarter of a period, Sumpolinsky et al., 1991, Young and Strogatz, 1999. This corresponds to 2s, which is 2 orders of magnitude longer than transhemispheric propagation delays. The arteriole vasomotion in each hemisphere is synchronous through the similarly delayed neuronal to vascular coupling, Figure 1. Go to sources of ultra-slow electrical signaling to couple to vasomotion. The theory of weakly coupled oscillators, Kuramoto, 1984, implies that vasomotor oscillations can phase lock to rhythmic neuronal activity within the same 0.1 Hz frequency band. We consider 3 such sources of activity, Figure 4a. The first 2 involve ultra-slow variations in neuronal electrical potential. One realization is in terms of an ultra-slowly varying envelope of high-frequency electrical oscillations in the brain, Leopold et al. 2003, Neer et al. 2008, Thompson et al. 2013. A second is in terms of potential ultra-slow variations in the electrocorticogram, ECOG, or electroencephalogram, e.g., he and Reichler, 2009. The ECOG and the EC measure spatially extended and temporally coherent extracellular current flow in the brain, Lemon, 1984. The third potential source of ultra-slow rhythmic activity is via neuromodulator nuclei. We consider the viability of each of these signaling mechanisms as a prelude to describing the locking of vasomotor activity to ultra-slow electrical signal in the brain. An external file that holds a picture, illustration, etc. Object name is NIMES 1634830F004.jpg. Figure 4. Ultra-slowly varying neuronal signals have multiple potential origins but are predominantly encoded by the ultra-slow modulation of the fast gamma band oscillations. A. Coupled oscillator model for resting state neurovascular coupling. The 3 potential coupling mechanisms for entrainment of vasomotor oscillations across separate regions of the brain. 1. The ultra-slow envelope of the gamma band oscillations shared across separate regions, as illustrated in panel B. 2. Ultra-slow electrical potentials per se that are shared across separate regions. And 3. Regional-specific neuromodulation of brain activity. Adapted from Mateo et al., 2017. B. A model to illustrate amplitude modulation using an ultra-slow signal to modulate a fast underlying oscillatory signal. The top trace is the product of a carrier centered at f-gamma equals 55 Hz and a 0.1 to 0.2 Hz band limited noise source for the ultra-slow modulate. The associated spectrum was computed with a bandwidth of half a Hz, black trace, or 0.05 Hz, blue trace. The bottom trace shows the frequency components of the ultra-slow envelope after demodulation and low-pass filtering to remove signals near 2 FAH. The associated spectrum was computed with a bandwidth of 0.05 Hz, from Mateo et al. 2017. C. Measurements and analysis of ultra-slow variations in the power of the surface ECOG during stage 2 sleep in a human subject, left panel, from Neer et al. 2008. The spectrogram of the ECOG shows the slow variation in power in the 40 to 90 Hz gamma band, left of center panel, window of 10s, bandwidth of 1.5 Hz, and color scales the logarithm of power from black/red, low, to white/yellow, high. The time series of the variation of integrated power in the gamma band of the ECOG shows oscillations on the 10s timescale. The spectrum of the derivative of this time series, a procedure that remove a power 1/2 trend from the spectrum, has statistically significant peaks at frequencies near 0.1 Hz. Reanalysis from Drew et al., 2008. Ultra-slow electrical signaling by modulation of gamma band oscillations. Gamma band oscillations refer to rhythmic brain activity in a broad range of frequencies, typically 30 to 80 Hz. It can be generated in vitro by networks of solely fast-spiking inhibitory neurons, Whittington et al., 1995, consistent with theoretical predictions, Hansel et al., 1993, Van Vreeswijk et al., 1994. In vivo, the details of the oscillatory waveform are shaped by the interactions between parvalbumin and pyramidal neurons, Cardin et al., 2009, Sahal et al., 2009, as well as by interactions with different classes of inhibitory neurons, White et al., 2000, and cholinergic modulation, Pinto et al., 2013. A key feature of gamma band oscillations is that the amplitude, or equivalently the spectral power, can vary over time. Leopold et al., 2003, Liu and Duan, 2013, Mateo et al., 2017, Neer et al., 2008, Thompson et al., 2013. In fact, an increase in power in the gamma band is a predictor of a subsequent increase in arteriole diameter and blood oxygenation. Mateo et al., 2017, Winder et al., 2017. Consistent with these physiological changes, there is a corpus of evidence that increased power in the gamma band correlates with an increase with the bold free signal. Gens and Logothetis, 2008, Keller et al., 2013, Lachaud et al., 2007, Lema et al., 2014. Niesing et al., 2005, Neer et al., 2007, Skalvink et al., 2010. While the power in the gamma band is the best predictor of an increase in arteriole diameter, the correlation with the spectral power extends down to lower frequencies. Gens and Logothetis, 2008, Mateo et al., 2017, Winder et al., 2017. As a matter of principle, amplitude modulation, Black, 1953, provides a means for ultra-slow signals to be transmitted in the brain, as can occur by modulating the interspike interval in a spike train by an inhomogeneous spike rate. Oken et al., 2019. Here, the gamma rhythm oscillations serve as the underlying carrier frequency, and the broadband, 0.1 Hz rhythm modulates the amplitude of the gamma rhythm oscillations, Figure 4b. The power spectrum of the gamma rhythm oscillations is broadened by the modulation, but still centered at a high frequency. There is no spectral power at the broadband, 0.1 Hz modulation, Figure 4b. Demodulation of the signal, which involves spectral mixing and can be accomplished by the threshold properties of neurons, Arons et al., 2002, retrieves the ultra-slow signal, Figure 4b. Recent data from the mouse accessory olfactory bulb implies that ultra-slow modulation can be quite strong in practice, see Tour et al., 2020. Evidence for encoding of ultra-slow oscillations by the modulation of gamma rhythm oscillations comes from studies on monkey, Leopold et al., 2003, and human subjects, Neer et al., 2008. The human study involved bilateral recordings of multi-unit spike waveforms and the ECOG from the parietal and temporal lobes of both cortices in preoperative patients. A spectrogram of the ECOG, acquired while a subject was in non-REM sleep, shows that the power in the gamma band is slowly modulated, figure 4c. The spectral power of the modulation in gamma band power, i.e., the so-called second spectrum, shows peaks at frequencies near 0.1 Hz, Drew et al., 2008, figure 4c. These modulations are present during both states of sleep and states of wakefulness. Is the modulation in gamma band power correlated across hemispheres, as would be expected if these signals relate to transhemispheric correlation in the bold fMRI signal, figure 2d, and the arteriole diameter, figure 3c? The spike times at spatially adjacent regions, as well as at functionally linked areas of opposing hemispheres, are not coincident. In contrast, there is significant coherence in the modulation of the spike rate and in the power of gamma band oscillations in the ECOG across sites in opposing hemispheres, Neer et al., 2008. During rest, there is a relatively large and positive correlation coefficient of 0.6 between ultra-slow ECOG signals from contralateral auditory areas, as compared with smaller correlation coefficients between signals from auditory and non-auditory areas. The stimulus-linked synchrony between the auditory areas in opposite hemispheres is likely mediated through callosal connections, Engel et al., 1991. The literature therefore suggests that 0.1 Hz signals are transmitted as modulation of the high-frequency gamma band rhythm, which is coherent across mirrored regions in the two hemispheres. A final concern is the spatial specificity of the ultra-slow modulation that forms correlated signals across hemispheres. This was addressed using wide-field optical imaging of the dorsal surface of cortex in transgenic mice that express functional indicators of intracellular calcium, Barson et al., 2019, Ma et al., 2016, Vani et al., 2017, or transmembrane voltage, Chan et al., 2015, Mahajirani et al., 2013. These indicators report the accumulation of spiking activity, which approximates rectification of neuronal activity above a threshold level, figure 4b, and thus reports the ultra-slow envelope of neuronal activity. While strong correlations occur for frequencies over the range from 0.1 Hz to 10 Hz, the amplitude of the oscillations across the dorsal surface is greatest in the 0.08 to 0.16 Hz frequency band, i.e., centered near 0.1 Hz, as opposed to bands below this frequency and up to 10 Hz, Vani et al., 2017. Further, bilateral symmetry of the neuronal response is largely reduced in acallosal mice, Mahajirani et al., 2010, albeit this issue is not without controversy, Quigley et al., 2003, Tishka et al., 2011. Ultra-slow changes in the electrocorticogram report non-neuronal dynamics. The EEG is traditionally recorded as surface potentials across the brain, Lemon, 1984. Ultra-slow variations in the ECOG or EEG, also referred to as DC potentials, are presumed to result from ultra-slow changes in active membrane currents, Besson et al., 1970, and are a complementary means to support ultra-slow neuronal activity to that of modulation of the high-frequency gamma rhythm, figure 4b, c. Yet extensive experimental evidence has shown that EEG signals with spectral components below 1 Hz have a non-neuronal origin. One mechanism to generate ultra-slow surface potentials is through changes in the volume of blood in the brain. The electrical potential of the blood is negative relative to that of the cerebrospinal fluid, Held et al., 1964, Sorensen and Severinghaus, 1970. Thus, dilation of arterioles, which in the awake animal can take place over seconds, and dilation of veins, which can occur over tens of seconds, Drew et al., 2011, Huo et al., 2015, Pissarro et al., 2013, will generate ultra-slow changes in the EEG. Besson et al., 1970, Nida et al., 2004, Van Hattaloa et al., 2003, Voipio et al., 2003, that are unrelated to neuronal activity per se. Experiments on human subjects have shown that a shift in blood volume, driven by changes either in posture or breathing that are unlikely to induce significant neuronal effects, will drive ultra-slow changes in the EEG. In the Muller maneuver, the subject attempts to inhale with their mouth closed and nostrils plugged. In the Valsalva maneuver, the subject attempts to exhale against a closed airway. This is a common means to equalize pressure to the sinuses. These procedures drive a change in blood volume, measured with near-infrared spectroscopy, NIRS, along with relatively large amplitude EEG signals that follow the change in blood volume, Van Hattaloa et al., 2003, Figure 5a. Tilting the head up or down, Figure 5a, as well as compression of the jugular vein give similar results, Van Hattaloa et al., 2003. Complementary experiments that involve vasodilation support the hypothesis that slow potentials are generated by a change in volume of the blood. In particular, inhalation of carbon dioxide causes vasodilation on the timescale of minutes, Besson et al., 1970, and Guy and Nguyen, 1996, and leads to concurrent changes in the EEG signal in the less than 1 Hz frequency range, Voipio et al., 2003. An external file that holds a picture, illustration, etc. Object name is NIMS1634830F005.jpg. Figure 5. Ultra-slowly varying extracellular potentials, i.e. DC potentials, can have a non-neuronal origin. A. 4 changes in human body position that alter cerebral blood volume, i.e. Muller and Valsalva maneuvers and head tilt maneuvers drive changes in EEG potential as measured with scalp electrodes. The change in total blood volume was found from near-infrared absorption of brain blood perfusion using scalp emitters and detectors, Ichikawa et al., 1999. The EEG scalp electrodes were at the 10 to 20 international coordinates CZ, T3 plus T4 for Muller and Valsalva, FZ, right mastoid for head up, and CZ, right mastoid for head down. From Van Hattaloa et al., 2003. B. Inhalation of CO2, a vasodilator, drives a large shift in ECOG potentials with no effect on the transmembrane potential or spike rate of a simultaneously recorded neuron. Intracellular and DC ECOG recording during recurrent spike wave seizures and hypoventilation in a cat under ketamine xylazine anesthesia, from Nida et al., 2004. The gold standard in these neurovascular studies is to record slow changes in the ECOG that are induced by vasodynamics concurrent with the electrical activity of nearby neurons. Such studies were performed with cats in which the CO2 content of the inhalation gas could be altered to induce vasodilation, Nida et al., 2004. Consistent with a vascular but not with a neuronal origin of ultra-slow electrical signals, simultaneous intracellular recordings from neurons, Figure 5b, and astrocytes show that the ultra-slow ECOG signals are reflected in neither the neuronal nor astrocyte membrane potential, Nida et al., 2004. Further, extracellular measurements as a function of depth in cortex failed to find a reversal of the polarity of the ultra-slow signals with cortical depth, inconsistent with the synaptic origin of these potentials, Nida et al., 2004. In toto, the data refute the notion that ultra-slow neuronal signals are present in ultra-slow brain-wide ECOG signals, as opposed to the modulatory signal of the high-frequency gamma band carrier seen in the ECOG. Are there other neuronal origins of ultra-slow electrical activity? Intracellular neuronal currents with variation in the 1 Hz range have been described, Surrogate et al., 1993, but there are no reports of slower ionotropic currents with the exception of nicotinic acetylcholine receptors that contain the alpha-7 and beta-2 receptor subunits. These subunits may play an important role in the generation of ultra-slow fluctuations in the spike rates of neurons in frontal cortex, Kucoli et al., 2016. Other ion channels show depolarization-induced changes in recovery times that last up to hundreds of seconds, Toibi et al., 1998. And spontaneous synaptic release shows correlations over a multiplicity of long timescales, Lowen et al., 1997. While currents that rely on reaction diffusion kinetics and concentration-dependent rates can be quite slow, they are not a priori oscillatory in nature. Targeted neuromodulatory input to cortex. There is evidence that slow signals from cholinergic modulatory centers provide brain-wide modulation of the gamma rhythm, Turch et al., 2018. The composite evidence also suggests that cholinergic signaling activates the M5 metabotropic pathway, Yamada et al., 2001, in penetrating arterioles, Adaki et al., 1992, Haught et al., 2013, Sato et al., 2001, Voucher and Hamill, 1995. Several studies have shown that projections from the basal forebrain, which contains a prominent cholinergic center, are targeted to specific brain regions. Kim et al., 2016a, Saper, 1984, Wu et al., 2014, Zaborski et al., 2015. In particular, cholinergic targeting is specific at least down to the level of primary sensory areas, Kim et al., 2016a, consistent with the murine projectomy of the basal forebrain, Li et al., 2018. Some cholinergic neurons could co-target functionally related areas, Li et al., 2018, though finer-scale organization like retinotopy may not be present, Hub-Gorgs et al., 2018. The inputs to basal forebrain neurons also show a targeted input from different brain regions, Gilo and Zaborski, 2017. The neuromodulator noradrenaline, released by projections from the locus coeruleus, also functions to change brain-wide behavior. Chemogenetic activation of the locus coeruleus leads to an increase in the functional connectivity, as determined from the pairwise cross-correlations among signals from different brain regions, Zerbe et al., 2019. However, unlike the case for basal forebrain input, the projections from locus coeruleus appear to be cortex-wide and diffuse, with no evidence for regional projections, Kim et al., 2016a. Of interest, noradrenaline release leads to global activation as a function of the cognitive task, Cardozo et al., 2019. We conclude that vasomotion could in principle phase lock to slow variations in neuromodulation. Only cholinergic modulatory input has the regional specificity that suggests feasibility, yet even for this case there is no evidence for changes in modulation on the short, 2 mm correlation length scale seen in rodents, He et al., 2018, Mateo et al., 2017. Nor is there published evidence for even broadband oscillations at the 0.1 Hz range. While we cannot dismiss neuromodulation as a candidate mechanism for locking vasomotion to neuronal activity, current evidence deems it untenable. Go to ultra-slow variations in gamma band power entrains vasomotion. The ability to measure electrophysiological quantities concurrent with vasodynamics is essential to decipher the biophysical basis of resting state fMRI signals, Vilringer et al., 2006. A plethora of past work has made progress with the use of mixed electrical and optical-based recording of neuronal activity concurrent with measures of blood flow, often both hemodynamics and vasodynamics in monkeys, Gens et al., 2012, Gens and Logothetis, 2008, Logothetis et al., 2012, Logothetis et al., 2001, Skalvink et al., 2010, Shemuel et al., 2006, Shemuel and Leopold, 2008, Rat, Du et al., 2014, He et al., 2018, Schwam et al., 2017, Thompson et al., 2014, and Mouse, Annenberg et al., 2014, Lee et al., 2010, Mott et al., 2016, Mateo et al., 2017, Pan et al., 2015, Schultz et al., 2012 et al., 2017, Wang et al., 2018, Winder et al., 2017. While mice have the great advantage of genetic introduction of functional labels, this can also lead to complications when the insertion of transgenes disrupt the coding sequence of endogenous genes, Goodwin et al., 2019, or labels lead to seizure activity, Steinmetz et al., 2017. The connection between vasomotor oscillations in pile arterioles and fluctuation in the envelope of the gamma band of the local field potential, LFP, from the superficial layers was established in mouse, Mateo et al., 2017. We first consider the temporal variation in the spectral power of the LFP in the upper layers of cortex in relation to changes in the diameter of the surface arterioles, Figure 6a. The field potential shows epochs of enhanced activity across all frequency bands. The variations in power are greatest in gamma band and, although episodic, are broadly distributed with a periodicity near 0.1 Hz. Crucially, changes in the diameter of surface arterioles covary with the power in the gamma band, Figure 6b. The timing of the fluctuations is such that the electrical activity leads that of the diameter by 2s both in this example, Figure 6c, and as an average over all observations. These data are consistent with the hypothesis that ultra-slow fluctuations in electrical activity drive changes in arteriole diameter. While the latency between increases in neuronal activity and vasodilation is 2s, that between the increase in neural activity and the bold fMRI signal is slightly longer, i.e. 3s, as a result of a delay of the increased oxygenated blood reaching the veins, Mateo et al., 2017, Figure 1a. To test if local neuronal activity is sufficient to entrain vasomotion, the neuronal activity in mice that express channel rhodopsin in layer 5b pyramidal neurons was driven by light whose intensity modulated by a 40 Hz gamma rhythm carrier frequency and a 0.1 Hz ultra-slow rhythmic envelope, Mateo et al., 2017. We observed that the envelope of the gamma band and the diameter of a nearby arteriole are phase-locked, with electrical activity leading vasodilation by the same 2s as observed under natural conditions, Figure 6c. These data confirm that ultra-slow modulation of high-frequency neuronal activity can entrain vasomotion. The possibility that variations in arteriole diameter drive electrical activity was addressed through the use of mice that express halorhodopsin in arteriole smooth muscle, Mateo et al., 2017. Illumination of an arteriole with activating laser light leads to an 0.2 times dilation, similar to the value seen during vasomotion, Drew et al., 2011. Crucially, driving dilation of the smooth muscle fails to lead to a change in the LFP and a large drop in the spectral coherence between the vessel diameter and the driven envelope of the electrical activity. These data support the interpretation that vasomotion does not drive aggregate neuronal activity. While the interaction between neuronal activity and vasomotion was unidirectional, an in vitro study has demonstrated feedback from blood vessels onto neuronal output, Kim et al., 2016b. Following pressurization of parenchymal arterioles, the vessels develop myogenic tone. A shift in the level of tone by challenging the arterioles with a change to higher pressure increases constriction, while lower pressures led to dilate. Importantly, upon an increase in pressure to induce vasoconstriction, pyramidal neurons suppressed their activity, while pressure-evoked dilation led to an increase in pyramidal neuron activity. The relation of this homeostatic feedback mechanism to neurovascular coupling remains an open issue. Fidgeting movements are a driver of ultra-slow signals. A potential contributor to the ultra-slow fluctuations in gamma band power are small motor movements of the face and limbs that are referred to as fidgeting, Drew et al., 2019, Winder et al., 2017. Studies with humans and primates have shown that blinking, Bristow et al., 2005, Gipponi et al., 2015, swallowing, Byrne et al., 1999, Hamdi et al., 1999, and small head motions, Yan et al., 2013, are accompanied by bilateral activation of motor and somatosensory areas of the brain. Fidgeting appear to be independent of external sensation, but rather is a consequence of non-volitional neuronal events. Fidgeting occurs at the rate of several events per minute, enabling these events to drive ultra-slow signals, Huo et al., 2014, Kaminer et al., 2011, Lear et al., 1965. Fidgeting can take place in absence of stimulation, Stringer et al., 2019, or accompany a learned task in mice, Musall et al., 2019, Sackhoff et al., 2019, even though it is irrelevant to the task per se. For example, pressing a bar is accompanied by movements of the face and forelimbs and protraction of the vibrissae. Of relevance to blood flow, the functional form of the coupling between neuronal activity and hemodynamic signals appears the same for dilations evoked by either fidgeting, sensory stimuli, or volition, Power et al., 2012, Winder et al., 2017, Figure 6d, FF. Fidgeting is activated by neuronal motor circuits and presumably drives reafferent or efferent inputs to somatosensory regions in cortex. Thus, in principle, fidgeting can drive vasodilation and entrain vasomotion. We obtained new data to characterize the relation of fidgeting to the entrainment of vasomotion in alert, head-fixed, wild-type mice. Following past methods, Winder et al., 2017, we recorded vibrissa movement, body movement, and concurrent changes in the diameter of pile arterioles, Figure 6d, experimental details in the legend. Epochs of spontaneous whisking and body motion are observed, the largest of which are clearly correlated with relatively large transient vasodilation events. As an average over mice and vessels, We observe that the movement precedes the vasodilation by 2s for both whisking and whole-body motion, figure 6e, similar to that for the gamma band power, figure 6c. Thus, fidgeting is a likely contributor to the source of ultra-slow neuronal activity, albeit this shifts the question to what neuronal or environmental source drives fidgeting. We performed a quantitative assessment of the contribution of body movement to the neuronal entrainment of vasomotion across our existing data with head-fixed mice, Mateo et al., 2017, figure 6ac. Previously unpublished accelerometer records of the motion of the body of the mouse were used to determine the contribution of fidgeting to vasomotion, figure 6f, experimental details in the legend. As an average over mice and vessels, body motion leads to transient dilation of the pile arterioles that rises over an interval of 2s, the same as that for stimulus-driven changes in arteriole diameter, figure 1a. The movement-related changes in diameter encompassed 0.27 of the significantly correlated changes between vessel diameter and the gamma band LFP, center figure 6f. Critically, fidgeting was correlated with the largest dilations, right figure 6f. In toto, fidgeting is a significant contributor to the neurological events that entrain vasomotion in mice. We suggest that fidgeting is likely to play a role in the origin of human resting state bold re-signals as well, Drew et al., 2019. The punctate nature of fidgeting events is consistent with the punctate, albeit loosely rhythmic nature of resting state activity in humans, Liu et al., 2018, Liu and Duin, 2013, Petridou et al., 2013, and fidgeting behaviors often involve bilateral actions, Drew et al., 2019, consistent with the bilateral nature of spontaneous activations, figure 2d. Fidgeting behaviors are likely to be associated with bold fMRI signals in regions outside sensory and motor areas of the brain, including areas activated by cognitive tasks, Drew et al., 2019. For example, swallowing activates the insula, Martin et al., 2001, and licking, Nakano et al., 2013, and head motion, Bright and Murphy, 2015, activate multiple brain regions. It is an open issue if the sensory motor processing that is associated with fine-scale self-motion is part of the ideation captured by resting state bold fMRI. Nonetheless, experimental designs exist that can flag and censor epochs of even modest self-motion in human subjects, Power et al., 2012, Siegel et al., 2014. Transitions among behavioral states may drive ultra-slow signals. A seemingly high proportion of the resting state bold fMRI studies in animals and humans did not closely monitor behavior or arousal. Thus, a wide variety of behavioral states may be sampled and averaged together. Head-fixed mice will sleep with their eyes open, Usges et al., 2018. So careful physiological monitoring of body motion, and pupil size is needed to ensure that the sleep state vasodynamics, Foltz et al., 2019, Horowitz et al., 2009, do not contaminate resting state measurements. Post-hoc examination of human connectome data has indicated many if not most subjects fall asleep during the scanning process, Taliazuki and Laufs, 2014. Sleep-to-wake transitions, Horowitz et al., 2009, Liu et al., 2018, may thus drive events that account for a substantial component of functional connectivity, Liu and Duin, 2013, Petridou et al., 2013. Go to mechanisms for correlated bilateral vasodynamics. The dominant feature of resting state connectivity is the bilateral symmetry across hemispheres, figure 2d. The details of bilateral coactivation can be explored via the coherence between changes in arteriole diameter for vessels in mirrored regions. In fact, precise, simultaneous measurements of vessel diameter show that arterioles in mirrored regions oscillate in synchrony with near-unity coherence, Mateo et al., 2017, figure 3c. The coherence is severely reduced in ankylosal mice, Mateo et al., 2017, consistent with the finding that bilateral neuronal signaling is similarly reduced in ankylosal mice, Mahajarani et al., 2010. Yet, as noted previously, the evidence for mirrored activation in ankylosal human subjects is equivocal. Vascular geometry that enhances bilateral symmetry. While the origin of symmetry in resting state connectivity is typically attributed to interhemispheric projections, there are other systemic vascular contributors. Fluctuations in the inflow of blood at the level of the left and right carotids are strongly coherent at 0.01 to 0.3 Hz, i.e., the range of vasomotor frequencies in awake rats, Revel et al., 2012. Arterial blood oxygenation also fluctuates with fluctuations in respiration rate and with the respiratory cycle, Zhang et al., 2019. These lead to delayed fluctuations in the global fMRI signal throughout the brain and into the sagittal sinus, Tong et al., 2019. Unfortunately, the fluctuations from the carotid supply tend to dephase as blood travels with different transit times through the brain. Thus, fluctuations in inflow and oxygenation cannot be completely removed by regression of a brain-wide signal, Tong et al., 2015. A further complication occurs since vasomotion will change the transit times. Thus, measures of blood oxygenation can be systematically biased if they occur at locations with different transit times as opposed to mirrored brain regions. For this reason, recent reports of small shifts in the timing of both fMRI signals in humans, Mitra et al., 2015, could be the result of small changes in the perfusion times or oxygenation of blood to different regions of the brain. As a clinical matter, this effect can highlight perfusion deficits after stroke, LV et al., 2012, and may explain how low-grade brain tumors show coherent oscillations in the bold fMRI with their putatively healthy contralateral brain regions, Gupta et al., 2018. Bilaterally synchronized modulatory inputs can drive bilateral oscillations. There are direct cholinergic cross-hemispheric connections between the basal forebrain and the two hemispheres, Carnes et al., 1990, Simba et al., 1988. These connections should not be affected in ankylosal animals, thus preserving common input to mirrored transhemispheric cortical regions. Activation of the rostral ventral lateral medulla, RVLM, a brainstem nucleus that sits on the ventral surface of the brain below the breathing center, Golanov et al., 1994, leads to an increase in cortical blood flow. This nucleus receives input from neighboring breathing centers, Dempsey et al., 2017, and will increase blood flow in response to a decrease in atmospheric PO2, Golanov and Race, 1996. The RVLM contains primarily noradrenergic neurons, Abbott et al., 2012. The limited anatomical studies published to date have not identified direct nor complete indirect projections from RVLM to the cortex, Card et al., 2006. Nonetheless, in one likely pathway, the RVLM projects to the medullary cerebral vasodilator area in brainstem, then projects to the subthalamic cerebral vasodilator area in the diencephalon, which incorporates zona incerta and the fields of Forel, and finally projects to deep layers of cortex, Golanov et al., 2001, Ilcha and Golanov, 2004. The specifics of the drive to cortical blood vessels is an open issue. Go to Technical sources of ultra-slow noise contributions in fMRI. As a means to reduce voluntary motion of the head, contemporary rodent resting state fMRI studies have been primarily performed on anesthetized head-fixed but free-breathing animals, Biswal and Konnerpati, 2009, Williams et al., 2010, Zhao et al., 2008. However, cardiac and respiratory-based artifacts in rodent resting state BOLD and CBV fMRI remain to be carefully identified and removed from the data. These difficulties stand in contrast to the significant efforts made for the human BOLD fMRI studies, Byrne et al., 2006, Dali et al., 1999, Glover and Lee, 2000, Hu et al., 1995, Shmueli et al., 2007, Wise et al., 2004. The adoption of signal processing methods developed for human studies, Caballero Gauds and Reynolds, 2017, Murphy et al., 2013, will be crucial to reduce the interference of cardiorespiratory artifacts with the spatiotemporal patterns of the resting state fMRI signals in animals, Abreu et al., 2017, Biswal et al., 1996, Bright and Murphy, 2017, Feinberg and Setzenpop, 2013, Kivinyami et al., 2005. Respiration-induced B0 offset motion. Magnetic resonance image acquisition relies on Larmor frequency-based spatial encoding through the sequence-specific magnetic field gradients across a subject. The homogeneity of the constant magnetic field, designated B0, determines the quality of magnetic resonance images, Durand et al., 2001. However, the subject inside the magnetic resonance scanner can continually disturb the homogeneity of the B0 field by movement of their body. Biswal et al., 1996, Frank et al., 2001, Lowe et al., 1998, Van de Mortelle et al., 2002. This includes fidgeting, figure 6D, as well as breathing and other physiological motions. In practice, breathing leads to an expanding and contracting B0 field near the brain, figure 7A. Thus, artifacts in bold MRI can originate from parts of the body that are far away from the field of view in the brain, figure 7B. An external file that holds a picture, illustration, etc. Object name is NIMES 1634830F007.jpg. Figure 7. The aliasing effect of the respiratory-induced magnetic field B0 distortion. A. Schematic drawing of the B0 field on free-breathing rats with different extents of chest movements and associated magnetic field lines. The distorted B0 field is caused by the large chest movement during the respiratory cycles. Red lines. Amber circles are pickup coils. From Pais-Roden et al., 2018. B. The coronal sections from a three-dimensional echoplanar imaging sequence acquired at 1.2s repetition time, TR, from 0 to 7.2s, from a spontaneously breathing anesthetized rat. These data show the effect of the B0-related motion artifacts at 0.16 Hz as the function of time, from Pais-Roden et al., 2018. C. The respiratory trace, black, chest movement, and the downsample time course, red line, for a 1-year sampling rate with TR equals 1.2s, upper panel. The power spectral density shows the aliased signal, peaked, Fres-1/TR, i.e. 0.16 Hz, of which the Fres is around 1.0 plus or minus 0.1 Hz. The fMRI time course and spectral density of a voxel in the brain, middle panel, or in a phantom, bottom panel, over the animal's head, shows the oscillatory signal with a bandwidth of 0.16 plus or minus 0.1 Hz, which is dominated by the respiration-induced B0 artifacts. The brain and phantom voxels are defined by the two arrows in panel D, from Pais-Roden et al., 2018. D. Map of the power at the artifact frequency, 0.16 plus or minus 0.1 Hz, shows the voxel-wise spatial correlation to the B0-related artifacts. The high power scales indicate the strong B0-related artifacts, from Pais-Roden et al., 2018. Methods to compensate for the time-dependent B0 field distortions are known. Raj et al., 2000, Van Gelderen et al., 2007. Yet, in contrast to studies on human subjects, the higher magnetic fields used for animal bold and CBV fMRI studies will exaggerate the effects of physiological contributions to an inhomogeneous B0 field. In addition, both arteriole CO2, Wise et al., 2004, and respiratory volume changes, Byrne et al., 2006, can cause variations in the resting state bold fMRI signals that lead to falsely interpreted signals. Aliasing of physiological rhythms. Rats have an approximately 5 to 7 Hz cardiac frequency and approximately 1 to 2 Hz respiration rates under anesthesia. The conventional echoplanar imaging protocol with rodents involves a 1 to 2s period of repetition, TR, or equivalently a sampling rate, denoted Fs, of Fs equals 1/TR equals 0.5 to 1.0 Hz. The low rate of sampling compared to physiological processes that distort the fMRI signal will cause aliasing of both cardiac and respiratory contributions down to frequencies that may also be erroneously associated with resting state dynamics. This is well described for human resting state fMRI studies, Biswal et al., 1996, Dali et al., 1999, De Luca et al., 2006, Frank et al., 2001, Lowe et al., 1998, and is seen in rat as well, Pais-Roden et al., 2018. For example, when the sampling rate is Fs equals 0.8 Hz and the spontaneous breathing rate of an anesthetized rat is Fb equals 1.0 plus or minus 0.1 Hz, the signal is aliased to Fb-Fs equals 0.2 plus or minus 0.1 Hz. This is illustrated by measuring breathing at Fs-Fb and observing the aliased rate that is seen when the data is downsampled to fs equals 0.8 Hz, figure 7c. Aliasing is observed in the fMRI signal of both brain and a phantom above the head of the animal when the sampling rate is fs equals 0.8 Hz, figure 7c. This illustrates the combined effect of distortion of the B0 field and aliasing to form a false image of a resting state signal, figure 7d. Fast fMRI methods clearly will avoid aliasing of both the cardiac and respiratory contributions to the fMRI signals from rodents, Williams et al. 2010, Yu et al. 2014. A similar fast sampling strategy has been well implemented to remove the physiological noise for fMRI measurements from human brains, Agrawal et al. 2020. However, it is much more difficult to avoid the higher frequency cardiac-specific aliasing artifacts, especially for awake rodent fMRI, Desai et al. 2011, Ferenczi et al. 2016, Liang et al. 2015. Further, cardiac and respiratory cycles can fluctuate individually and in a coupled manner at frequencies close to the 0.1 Hz frequency range for ultra-slow signals. These fluctuations can serve as additional confounding sources for resting state Boltzmann studies, Shmueli et al. 2007. Go to conclusions. Motivated by a desire to understand the biophysical basis of resting state fMRI, figure 2, we have reviewed data on the relation of spontaneous changes in blood flow and blood oxygenation in the brain to changes in the underlying neuronal activity, figure 8a. Ultra-slow vasomotor oscillations occur in the smooth muscle cells that encircle brain arterioles and lead to rhythmic changes in the diameter of arterioles within a frequency band near 0.1 Hz, figure 3. Further, collective neuronal dynamics in the forebrain lead to the generation of high-frequency gamma-band oscillations whose amplitude is modulated at the ultra-slow rate of 0.1 Hz, figure 6b. These ultra-slow neuronal oscillations will entrain the arteriole oscillations to link brain vasodynamics with non-volitional neuronal activity, fig 6ac. The entrainment is statistically significant, figure 6, even though the vasculature also responds to common, cortex-wide inputs and homeostatic physiological inputs. The entrainment of vasomotion by neuronal spiking provides a means for arterioles in regions of the brain that are separated, yet connected through callosal projections to have synchronous vasomotor activity, figure 3c. Within the technological framework of fMRI, vasomotor activity can be measured through changes in arteriole volume, i.e. CBV fMRI, or changes in blood oxygenation secondary to changes in diameter, i.e. BOLD fMRI. The flow diagram of figure 8a formalizes this description. An external file that holds a picture, illustration, etc. Object name is NIMES1634830F008.jpg. Figure 8. Coupled oscillator model for resting state neurovascular coupling. A. Interactions for one region. Variations in gamma-band electrical power lead to partial entrainment of the vasomotor oscillations in the smooth muscle of cortical surface and penetrating arterioles. Increases in gamma-band electrical power dilate the arterioles and lead to an increase in the supply of fresh blood, as measured by CBV. The increase in diameter subsequently leads to an increase in blood oxygenation and a positive change in the Boltzmann signal. Coupling can be via callosal projections or via common input. B. Circuit of the pile arteriole network in terms of coupled smooth muscle oscillators. Each oscillator represents the activity of all muscles in one arteriole. The oscillators communicate with one another via the hexagonal lattice of electrically joined endothelial cells that form the lumen, figure 6. Oscillators in a given region of the brain may also receive electrical input from underlying neuronal oscillators. This input can dominate and lead to a parcellation of the frequency of vasomotor oscillations, as observed by fMRI, figure 2b. Vasomotion is synchronous within a given region of the brain yet can occur with different frequencies in different regions, for example, labeled blue and red for regions 1 and 2 respectively, and labeled green for an intermediate region without neuronal drive. Significance of the ultra-slow coupling. Functional MRI allows one to decode the neural activity in the brain to the extent that neural activity is represented by bold signals, K et al., 2008. A key issue is how well hemodynamic signals report neural activity. In Supplemental Table 1, we list the correlation coefficient R2, the so-called variance explained, between neural activity and either the bold or CBV fMRI or equivalent optical signal. Supplemental Text Box 1 relates R2 to the spectral coherence, CF. In all of these studies, there was a highly significant correlation between the hemodynamic response and neural activity. This means that resting state bold fMRI signal or equivalent are, to varying extents, driven by neural activity. Yet the value of 2 South African rand is universally less than 0.5 and typically around 0.1, Skalvink et al., 2010, Shemuel and Leopold, 2008. This means that only 0.1 to 0.5 of the hemodynamic signal is related to neural activity. A value of 2 South African rand equals 0.5 between gamma band neuronal activity and the simultaneously recorded bold fMRI signal occurs for visual stimulus evoked activity in the UN anesthetized primate, Gens and Logothetis, 2008. Given the strong drive of visual stimuli, this suggests that the value 2 South African rand equals 0.5 seen in rest state measurement is likely to be an upper bound. If we further consider that fidgeting occurs 0.3 of the time as an upper bound, we expect that at the most 0.5 by 0.7, 0.4 of the resting state bold fMRI signal is related to internally driven brain dynamics. Thus, resting state bold fMRI, as currently practiced, provides unique capabilities to infer connectivity in the human brain, yet is a technique with an inherently low signal-to-noise ratio. The role of vasomotion. What is the normal physiological purpose served by the ultra-slow vasomotor oscillations of brain arterioles? Recent work points to the necessity of vasomotion for the clearance of large solutes, Van Vliet et al., 2020, principally through the perivascular space between the smooth muscle and endothelial cells, Chair and Ostrich, 1974, Reynolds et al., 1985. These solutes include beta-amyloid, Iliffe et al., 2012. To the extent that vasomotion is entrained by the envelope of the gamma band power, Mateo et al., 2017, Figure 6, it is of interest whether increases in gamma power in the brain through sensory stimulation also increases the clearance of beta-amyloid, Yaccarino et al. The ability of vasomotion to drive solutes must depend on a mechanism to set the direction of flow, such as peristaltic motion, Aldea et al., 2019, or yet to be discovered valves in the perivascular space, Mateo et al., 2010. Next steps. The nature of the local neuronal circuitry that generates ultra-slow signals and modulates the gamma rhythm remains an open issue. In particular, the exact frequency of ultra-slow signals varies across the brain with millimeters to centimeters of coherence, Mitra et al., 1997, Vani et al., 2017, Figure 2b. Such spatially restricted modulation could be accomplished by even small changes in the activation of currents in individual cells that contribute to generate the gamma rhythm, Copel and Lemasson, 1994. In this way, the ultra-slow signals are purely modulatory, Figure 4, and the ECOG will not contain ultra-slow signals, Figure 5. Recent work describes a fast mechanism to transform subsurface microvasculature signaling into changes in arteriole diameter, Longden et al., 2017, Figure 1c. Yet the nature of electrical signaling across the lattice of pile surface vasculature remains to be solved, Hillman, 2014. At a phenomenological level, the pile arteriole oscillators form 2 sets of interactions, Figure 8b. On the one hand, each arteriole is directly connected with 4 neighboring oscillators, Blinder et al., 2010, via gap junctions between endothelial cells, Figure 1c. This connectivity acts to frequency lock vasomotion across all vessels across the surface, Sakaguchi et al., 1988, Strogatz and Marola, 1988. On the other hand, underlying neurons can drive the local pile vessels at different frequencies. This should break the coherence of vessels across the cortical mantle so that the vessels parcellate into regions with different vasomotor center frequencies, Figure 8b. Bold fMRI data from humans support such a parcellation on the scale of tens of millimeters, Figure 1b. In fact, such parcellation has been used to segment different regions in cortex, Fox et al., 2005, Glasser et al., 2016. Animal models, together with large-field, high-precision optical imaging, Sofroniou et al., 2016, Tsai et al., 2015, should allow us to assess the phenomenology and the biophysics for this parcellation. A more accurate and high-resolution measure of functional connectivity may be obtained through measurements of CBV rather than fMRI, He et al., 2018. When the spatial resolution of fMRI can distinguish individual vessels that penetrate the cortex of the rat brain, Moon et al., 2013, Poplawski et al., 2019, Yu et al., 2012, the bold fMRI signal is located mainly at venual voxels, while the CBV fMRI signal is detected primarily at arteriole voxels, He et al., 2018, Yu et al., 2016. Thus, the single-vessel fMRI method will enable detection of the spatial correlation patterns of the resting state signal with vascular specificity, as seen optically in restricted regions, Mateo et al., 2017, Figure 3c. From a technical perspective, the spatial localization of single-vessel fMRI decouples the resting state signal from respiration-induced B0-related motion artifacts, Figure 7. The ability to resolve individual penetrating vessels with CBV fMRI provides a critical platform to explore the ultra-slow oscillatory patterns of vasodynamics across the brain. This is particularly important for understanding how pairwise interactions among different brain regions may lead to the great multitude of default networks of rodent, Chan et al., 2015, Ma et al., 2016, Mahajirani et al., 2013, Mahajirani et al., 2010, and human, Gressius et al., 2003, Reichler et al., 2001, Sporns et al., 2005, brain activity. Finally, as a practical matter, experimenters should monitor behavior and arousal state as much as possible to differentiate between activity generated during spontaneous movements, sleep, and activity generated during true rest, Drew et al., 2019. Monitoring of behavioral state has become de rigueur in systems neuroscience experiments using awake rodents and primates, Egner and Branson, 2016, Wishaw and IQ Wishaw, 2014, makes use of standard equipment for studies with rodents, Kernikova et al., 2017, Mathis et al., 2018, Reimer et al., 2014, and should be adopted by the human fMRI community as well. Thus, it may be possible to use behavioral event markers to segregate resting state epochs with fidgeting from those without these movements and presumably increase the sensitivity to purely intrinsically driven brain-wide changes in bold or CBV fMRI signals. Go to Supplementary Material. Supplemental information for Drew et al. Click here to view 900K PDF. Go to Acknowledgements. We thank David Boas, Thomas Bragani, Jean-Pierre Chainju, Karishma Kabria, Anna Devore, Jessica Filosa, Parta Mitra, Mark Nelson, Jonathan Polimeni, Bruce Rosen, and Massimo Vergasola for valuable discussions. We thank Beth Friedman for comments on drafts of this manuscript. This work was supported by grants EB 02-17003, MH 114224, and NS 078168 to PJD, MH 111438, and NS 097265 to DK, NS 113278 to Xi and the Max Planck Society. Go to References: Alkjer C., Boetger D., and Machkov V., 2011. Vasomotion, What is Currently Thought? Active Physiology 202, 253-269. Alkar C. and Nielsen H. 2005. Vasomotion, Cellular Background for the Oscillator and for the Synchronization of Smooth Muscle Cells. British Journal of Pharmacology 144, 605-616. PMC Free Article. Abbott S.B., Kanbar R., Bokarishvili G., Coates M.B., Stornetta R.L., and Guillanet P.G. 2012. C1 neurons excite locus coeruleus and A5 noradrenergic neurons, along with sympathetic outflow in rats. Journal of Physiology 590, 2897-2915. PMC Free Article. Abreu R., Nunes S., Leal A., and Figueredo P. 2017. Physiological noise correction using ECG-derived respiratory signals for enhanced mapping of spontaneous neuronal activity with simultaneous fMRI. Neuroimage 154, 115 to 127. Adaki T., Baramides D.G., and Sato A., 1992. Stimulation of the nucleus basalis of Maynard increases cortical cerebral blood flow without influencing diameter of the pile artery in rats. Neuroscience Letters 143, 173 to 176. Agrawal U., Brown N., and Lewis L.D., 2020. Model-based physiological noise removal in fast fMRI. Neuroimage 205, E116231. PMC free article. Ahrens K.F., Levine H., Sewell H., and Kleinfeld D., 2002. Spectral mixing of rhythmic neuronal signals in sensory cortex. Proceedings of the National Academy of Sciences, USA 99, 15,176 to 15,181. PMC free article. Aldea R., Weller Rowe, Wilcock D.M., Carrare Rowe, and Richardson G., 2019. Cerebrovascular smooth muscle cells as the drivers of intramural periarterial drainage of the brain. Frontiers in Aging Neuroscience 11, E1. PMC free article. Annenberg E., Osteikitis P., Niitsu Y., Harrison T.C., Boyd J.D., Hilton B.J., Tetzlaff W., and Murphy T.H., 2014. Ministrokes in channelrhodopsin 2 transgenic mice reveal widespread deficits in motor output despite maintenance of cortical neuronal excitability. Journal of Neuroscience 34, 1094 to 1104. PMC free article. Atwell D., Buchan A.M., Charpak S., Lauritsen M., McVicker B.A., and Newman E.A. 2010. Glial and neuronal control of brain blood flow. Nature 468, 232 to 243. PMC free article. Bondatini P.A., Wong E.C., Hinks R.S., Tukovsky R.S., and Hyde J.S., 1992. Time course epi of human brain function during task activation. Magnetic resonance in medicine 25, 390 to 398. Barson D., Hamodi Az, Shen X, Lur G, Constable R.T., Cardin Ja, Crair M.C., and Higley M.J., 2019. Simultaneous mesoscopic and two-photon imaging of neuronal activity in cortical circuits. Nature Methods 16, 1-7. PMC free article. Beliveau J.W., Kennedy D.N., McKinstry R.C., Buckbinder B.R., Byskoff R.M., Cohen M.S., Viva J.M., Brady T.J., and Rosen B.R., 1991. Functional mapping of the human cortex using magnetic resonance imaging. Science 254, 716 to 719. Besson J.M., Woody C.D., Leonard P., Thompson H.K., Albie Fesser D., and Marshall W.H., 1970. Correlations of brain DC shifts with changes in cerebral blood flow. American Journal of Physiology 218, 284 to 291. Byrne R.M., Bondatini P.A., Cox R.W., and Shaker R., 1999. Event-related fMRI of tasks involving brief motion. Human Brain Imaging 7, 106 to 114. PMC free article. Byrne R.M., Diamond J.B., Smith M.A., and Bondatini P.A., 2006. Separating respiratory variation-related neuronal activity-related fluctuations in fluctuations from fMRI. Neuroimage 31, 1536 to 1548. Biswal B., Dio E.A., and Hyde J.S., 1996. Reduction of physiological fluctuations in fMRI using digital filters. Magnetic resonance in medicine 35, 107 to 113. Biswal B., Yetkin F.Z., Houghton V.M., and Hyde J.S., 1995. Functional connectivity in the motor cortex of resting human brain using echoplanar MRI. Magnetic resonance in medicine 34, 537 to 541. Biswal B.B. and Konnerpati S.S., eds. 2009. Resting state functional connectivity in animal models. Modulations by exsanguination, Springer protocols. PMC free article. Biswal B.B., Martin Min M., Zuo X. N., Goal S., Kelly C., Smith S.M., Beckman C.F., Adelstein J.S., Buckner R.L., Stan Colcomb S. et al., 2010. Toward Discovery Science of Human Brain Function. Proceedings of the National Academy of Sciences, USA 10, 4734 to 4739. PMC free article. Black H.S., 1953. Modulation Theory, New York, Van Nostrand. Blinder P., Shi A., Rarfi C.A., and Kleinfeld D., 2010. Topological Basis for the Robust Distribution of Blood to Rodent Neocortex. Proceedings of the National Academy of Sciences, USA 107, 12670 to 12675. PMC free article. Boland J.W., Wu C., Barbas H., Bokil H., Bota M., Brighter H.C., Klein Height, Doyle J.C., Freed P.J., Greenspan R.J., et al., 2009. A proposal for a coordinated effort for the determination of brain-wide neuroanatomical connectivity in model organisms at a mesoscopic scale. Public Library of Science, Computational Biology 5, E100334. PMC free article. Branscombe L.M., 1995. Confessions of a Technophile, American Institute of Physics. Bright M.G. and Murphy K., 2015. Is fMRI noise really noise? Resting state nuisance regressors remove variance with network structure. Neuroimage 114, 158 to 169. PMC free article. Bright M.G. and Murphy K., 2017. Cleaning up the fMRI time series, mitigating noise with advanced acquisition and correction strategies. Neuroimage 154, 1 to 3. Bristow D., Frith C., and Reese G., 2005. Two distinct neural effects of blinking on human visual processing. Neuroimage 27, 136 to 145. Buxton Airbay, 2001. The elusive initial dip. Neuroimage 13, 953 to 958. Caballero God C. and Reynolds R.C. 2017. Methods for cleaning the bold fMRI signal. Neuroimage 154, 128 to 149. PMC free article. Caesar K., Akgoran N., Matheson C., and Lauritsen M., 1999. Modification of activity-dependent increases in cerebellar blood flow by extracellular potassium in anesthetized rats. Journal of Physiology 520, 281 to 292. PMC free article. Card J.P., Sved J.C., Craig B., Rizada M., Vasquez J., and Sved A.F. 2006. Efferent projections of rat rostroventrolateral medulla C1 catecholamine neurons. Implications for the central control of cardiovascular regulation. Journal of Comparative Neurology 499, 840 to 859. Cardin Jha, Carlin M., Melitis K., Noblik U., Jong F., Diceroth K., Tsai L.H., and Moore C., 2009. Driving fast spiking cells induces gamma rhythm and controls sensory responses. Nature 459, 663 to 667. PMC free article. Cardozo MMB, Lima B., Saratin E.B., and Das A. 2019. Task-related hemodynamic responses are modulated by reward and task engagement. Public Library of Science, Biology 17, E30080. PMC free article. Carnes K.M., Fuller T.A., and Price J.L., 1990. Sources of presumptive glutamatergic/aspartatergic afferents to the magnocellular basal forebrain in the rat. Journal of Comparative Neurology 302, 824 to 852. Colley B. and Hamill E. 2010. Revisiting the Role of Neurons in Neurovascular Coupling. Frontiers of Neuroenergetics 9, E009. PMC free article. Chan O. Mahajarani M.H. Ledoux J.W., Wang and Murphy Th. 2015. Mesoscale infraslow spontaneous membrane potential fluctuations recapitulate high-frequency activity cortical motifs. Nature Communications 6, 7738, PMC free article. Chair H.F. and Ostrich L.H., 1974. Bulk flow of interstitial fluid after intracranial injection of blue dextran 2000. Experimental Neurology 45, 50-60. Dacey R.G. Jr. and Dueling B.R., 1984. Effect of norepinephrine on penetrating arterioles of rat cerebral cortex. American Journal of Physiology, Heart and Circulatory Physiology, 246, 380-385, Dolly M.S., Njim J., and Haxby J.V., 1999. Localization of cardiac-induced signal change in fMRI. Neuroimage 9, 407-415, De Luca M., Beckman C.F., DiStefano N., and Matthews P.M., 2006. fMRI. The resting state networks define distinct modes of long-distance interactions in the human brain. Neuroimage 29, 1359-1367, De Luca M., Smith S., DiStefano N., Federico A., and Matthews P.M., 2005. Blood oxygenation level-dependent contrast resting state networks are relevant to functional activity in the neocortical sensory motor system. Experimental Brain Research 167, 587-594, Dempsey B., Le S., A.T., Bokenyek P., Ramadas R., Jolly J.G., Minuet C., Neve R., Allen A.M., Goodchild A.K., et al., 2017. Mapping and analysis of the connectome of sympathetic premotor neurons in the rostral-ventrolateral medulla of the rat using a volumetric brain atlas. Frontiers in Neural Circuits 11, E9. PMC free article. Desai M., Khan I., Noblik U., Bernstein J., Atala H., Yang A., Kopel N., Buckner R.L., Graybiel A.M., Moore C., et al., 2011. Mapping brain networks in awake mice using combined optical neural control and fMRI. Journal of Neurophysiology 105, 1393-1405, PMC free article. Drew P.J., Duin J.H., Golanov E., and Kleinfeld D. 2008. Finding Coherence in Spontaneous Oscillations. Nature Neuroscience 11, 991-993. Drew P.J., Shi A., Driscoll J.D., Knudsen P.M., Davalos D., Blinder P., Akasaglu K., Tsai P.S., and Kleinfeld D., 2010. Chronic optical access through a polished and reinforced thin skull. Nature Method 7, 981-984. PMC free article. Drew P.J., Shi A., and Kleinfeld D. 2011. Fluctuating and sensory-induced vasodynamics in rodent cortex extends arteriole capacity. Proceedings of the National Academy of Sciences, USA 108, 8473-8478. PMC free article. Drew P.J., Winder at, and Zhang Q. 2019. Twitches, blinks, and fidgets, important generators of ongoing neural activity. The Neuroscientist 25, 298-313, PMC free article. Ducey, Volkov N.D., Koretsky A.P., and Pan Y. 2014. Low-frequency calcium oscillations accompany deoxyhemoglobin oscillations in rat somatosensory cortex. Proceedings of the National Academy of Sciences, USA 111, 4677-4686. PMC free article. Durand E., Van de Mortelle P.F., and Pacat-Cloire M., 2001. Artifact due to BO fluctuations in fMRI. Correction using the K-space central line. Magnetic resonance in Medicine 48, 198-201. Egner Sayre and Branson K. 2016. Computational analysis of behavior. Annual Review of Neuroscience 39, 217-236. Engel A., Koenig P., Kreider A.K., and Singer W., 1991. Interhemispheric synchronization of oscillatory neuronal responses in cat visual cortex. Science 252, 1177-1179. Feinberg D.A. and Setsenpop K. 2013. Ultra-fast MRI of the human brain with simultaneous multi-slice imaging. Journal of Magnetic Resonance 229, 90-100. PMC free article. Ferenczi E.A., Zalokowski K. Liston C., Groznik L., Warden Mr., Amatya D., Katovich K., Mehta H., Patnaud B., Ramakrishnan C., et al., 2016. Prefrontal cortical regulation of brain-wide circuit dynamics and reward-related behavior. Science 351, E9698. PMC free article. Ferezu I., Heiss F., Gentet L.J., Aronoff R., Weber B., and Peterson CCH, 2007. Spatiotemporal dynamics of cortical sensory motor integration in behaving mice. Neuron 56, 907-923. Filosa Ja, Bonif A.D., Straub S.V., Meredith Al., Wilkerson M.K., Aldrich R., and Nelson Mount, 2006. Local potassium signaling couples neuronal activity to vasodilation in the brain. Nature Neuroscience 9, 1397-1403. Fox M.D. and Reichler Me, 2007. Spontaneous fluctuations in brain activity observed with functional magnetic resonance imaging. Nature Reviews of Neuroscience 8, 700-711. Fox M.D., Snyder Oz, Vincent J.L., Corbetta M., Van Essen D.C., and Reichler Me, 2005. The human brain is intrinsically organized into dynamic, anti-correlated functional networks. Proceedings of the National Academy of Sciences, USA 102, 9673-9678. PMC free article. Fox P.T. and Lancaster J.L., 2002. Mapping Context and Content, the Brain Map Mode. Nature Reviews of Neuroscience 3, 319-321. Frank L.R., Buxton Airbay and Wong E.C., 2001. Estimation of respiration-induced noise fluctuations from undersampled multislice fMRI data. Magnetic Resonance in Medicine 45, 635-644. Fultz Ni, Bunmasser G., Setsenpop K., Stickgold R.A., Rosen B.R., Alamany Jr., and Lewis L.D., 2019. Coupled electrophysiological, hemodynamic, and cerebrospinal fluid oscillations in human sleep. Science 366, 628-631. PMC free article. Gao Ye, Ma Y, Zhang Q, Winder at, Liang Z, Antonori L, Drew P.J., and Zhang N, 2017. Time to Wake Up, Studying Neurovascular Coupling and Brain-Wide Circuit Function in the UN Anesthetized Animal. Neuroimage 153, 382-398. PMC free article. Gilo Mr. and Zaborski L, 2017. The Input-Output Relationship of the Cholinergic Basal Forebrain. Cell Reports 18, 1817-1830. PMC free article. Glasser M.F., Colson T.S., Robinson E.C., D.H.C., Harwell J., Yacoub E., Ugerbil K., Anderson J., Beckman C.F., Jenkinson M., et al., 2016. A Multimodal Parcellation of Human Cerebral Cortex. Nature 536, 171-178. PMC free article. Glover G.H. and Lee T.Q., 2000. Image-based method for retrospective correction of physiological motion effects in fMRI. Retrowicker. Magnetic Resonance in Medicine 44, 162-167. Gintz J., Merkel H., Nikos K., and Logothetis N.K., 2012. High-resolution fMRI reveals laminar differences in neurovascular coupling between positive and negative bold responses. Neuron 76, 629-639. PMC free article. Gintz JBM and Logothetis N.K., 2008. Neurophysiology of the bold fMRI signal in awake monkeys. Current Biology 18, 631-640. Golanov E.V., Christensen R.C., and Race D.J., 2001. Neurons of a limited subthalamic area mediate elevations in cortical cerebral blood flow evoked by hypoxia and excitation of neurons of the rostral ventrolateral medulla. Journal of Neuroscience 21, 4032 to 4041, PMC free article, Golanov E.V., and Race D.J., 1996. Contribution of oxygen-sensitive neurons of the rostral ventrolateral medulla to hypoxic cerebral vasodilatation in the rat. Journal of Physiology 495, 201 to 216. PMC free article. Golanov E.V., Yamamoto S., and Race D.J., 1994. Spontaneous waves of cerebral blood flow associated with a pattern of electrocortical activity. American Journal of Physiology, Regulatory, Integrative and Comparative Physiology 266, 204 to 214. Goodwin Lowe, Splinter E., Davis T.L., Urban R., He H., Braun Re., Chesler Ij., Kumar V., Van Minden and Jukum J. et al., 2019. Large-scale discovery of mouse transgenic integration sites reveals frequent structural variation and insertional mutagenesis. Genome Research 29, 494 to 505, PMC free article, Ghazi A. and Schwartz A.J., 2016. Large-scale functional connectivity networks in the rodent brain. Neuroimage 127, 496 to 509. Grecius M.D., Krosno B. and Rice al., 2003. Functional connectivity in the resting brain, a network analysis of the default mode hypothesis. Proceedings of the National Academy of Sciences, USA 100, 253 to 258. PMC free article. Grecius M.D., Supikar K., Menon V., and Doherty R.F., 2009. Resting state functional connectivity reflects structural connectivity in the default mode network. Cerebral cortex 19, 71 to 78. PMC free article. Grinvold A., Frostick R.D., Leek E., and Hildesheim R., 1988. Optical imaging of neuronal activity. Physiological Review 68, 1285 to 1366. Grinvold A., Frostick R.D., Siegel R.M., and Bartfeld E., 1991. High-resolution optical imaging of functional brain architecture in the awake monkey. Proceedings of the National Academy of Sciences, USA 88, 11559 to 11563. PMC free article. Giponi O., Odwar S., Pinid S., Wardak C., and Hamed S.B., 2015. fMRI cortical correlates of spontaneous eye blinks in the non-human primate. Cerebral cortex 25, 2333 to 2345. Gupta L., Gupta R.K., Postma A.A., Sahu P., Gupta Park, Patter R., Alawat S., Saha I., and Bax W.H. 2018. Advanced and amplified bold fluctuations in high-grade gliomas. Journal of Magnetic Resonance Imaging 47, 1616 to 1625. Haddock Re. and Hill C.E. 2005. Rhythmicity in arterial smooth muscle. Journal of Physiology 566, 645 to 656. PMC free article. Hamdi S., Rothwell J.C., Brooks D.J., Bailey D., Aziz Q., and Thompson D.G., 1999. Identification of the cerebral loci processing human swallowing with H215O PET activation. Journal of Neurophysiology 81, 1917 to 1926. Hansel D., Motto G., and Monnier C., 1993. Phase dynamics for weakly coupled Hodgkin-Huxley neurons. Europhysics Letters 23, 367 to 372. Harris of, Longden T.A., DeBertrand F., Hill Eubanks D., and Nelson Mount, of 2018. Endothelial GQ-PCR activity controls capillary electrical signaling and brain blood flow through PIP2 depletion. Proceedings of the National Academy of Sciences, USA 115, 3569 to 3577. PMC free article. He B.J. and Reichler Me, 2009. The fMRI signal, slow cortical potential in consciousness. Trends in Cognitive Sciences 13, 302-309. PMC free article. He Y, Wang M, Chen X, Pullman R, Polimeni Jr., Scheffler K, Rosen Br, Kleinfeld D, and UX 2018. Ultra-slow single-vessel bold and CBV-based fMRI spatiotemporal dynamics and their correlation with neuronal intracellular calcium signals. Neuron 97, 925-939. PMC free article, Held D, Finkel V, and RPJ, 1964. Electrical properties of cerebral spina, fluid. Journal of Neurophysiology 27, 942-959. Hillman E.M. 2014. Coupling mechanism and significance of the bold signal, a status report. Annual Review of Neuroscience 37, 161-181. PMC free article. Honey C.J., Sporns O., Camoon L., Gigonde X., Theron J.P., Muley R., and Hagman P. 2009. Predicting Human Resting State Functional Connectivity from Structural Connectivity. Proceedings of the National Academy of Sciences, USA 106, 2035-2040. PMC free article. Horowitz S.G., Fukunaga M., De Zwart J., Van Gelderen P., Fulton S.C., Balkin T.J., and Doohan J.H., 2009. Low-frequency bold fluctuations during resting wakefulness and light sleep, a simultaneous EEG fMRI study. Human Brain Mapping 29, 671-682. PMC free article. Hata H., Masamoto K., Uchida S., Sekiguchi Y., Takua H., Kawaguchi H., Shigemoto K., Sudo R., Tanishida K., 2H et al., 2013. Layer-specific dilation of penetrating arteries induced by stimulation of the nucleus basalis of Maynard in the mouse frontal cortex. Journal of Cerebral Blood Flow and Metabolism 33, 1440-1447. PMC free article. Hu XP, Liu TH, Parrish T, and Erhard P. 1995. Retrospective estimation and correction of physiological fluctuation in functional MRI. Magnetic Resonance in Medicine 34, 201-212. Huber L., Handwerker D.A., Jangraw D.C., Chen G., Hall A., Stuber C., Gonzalez Castillo J., Ivanov D., Merritt S., Guidi M. et al., 2017. High-resolution CBV fMRI allows mapping of laminar activity and connectivity of cortical input and output in human M1, Neuron 96, 1253-1263. PMC free article. Huo BX, Gao Yer, and Drew PJ, 2015. Quantitative separation of arterial and venous cerebral blood volume increases during voluntary locomotion. Neuroimage 105, 369-379. PMC free article. Huo BX, Smith JB, and Drew PJ, 2014. Neurovascular coupling and decoupling in the cortex during voluntary locomotion. Journal of Neuroscience 34, 10,975-10,981. PMC free article. Hoop Gorgs F., Kareem Jagaik K., and Voucher E. 2018. Topographic organization of cholinergic innervation, from the basal forebrain to the visual cortex in the rat. Frontiers of Neural Circuits 12, E0019. PMC free article. Yacarino H.F., Singer A.C., Martorell A.J., Rudenko A., Gao F., Gillingham T.Z., Mathis H., Seo J., Kritsky O., Abdurab F. et al., 2016. Gamma frequency entrainment attenuates amyloid load and modifies microglia. Nature 540, 230-235. PMC free article. Iodecola C. 2004. Neurovascular regulation in the normal brain and in Alzheimer's disease. Nature Reviews of Neuroscience 5, 347-360. Ichikawa N., Fujiwara M., Kawaguchi F., Kaga M., and Kawasaki I.S., 1999. Development of Optical Topography System ETG 100. DEDIX Report 34, 47-52. Ilcha CP and Golanov EV, 2004. Cerebrovasodilation evoked by stimulation of subthalamic vasodilator area and hypoxia depends upon the integrity of cortical neurons in the rat. Neuroscience Letters 368, 92-95, Ilif J.J., Wang M., Liao Y., Plog B.A., Ping W., Gunderson G.A., Benveniste H., Vates G.E., Dean R., Goldman S.A., et al., 2012. A paravascular pathway facilitates CSF flow through the brain parenchyma and the clearance of interstitial solutes, including amyloid beta. Science Translational Medicine 4, E111, PMC Free Article, Intaglietta M., 1990. Vasomotion and flow motion, Physiological Mechanisms and Clinical Evidence. Reviews in Vascular Medicine 1, 101-112. Powered J.D., Barnes Ka, Snyder Oz, Schlager B.L., and Peterson S.C., 2012. Spurious but systematic correlations in functional connectivity MRI networks arise from subject motion. Neuroimage 59, 2142-2154, PMC Free Article. Quigley M.A., Quartz D., Tursky P.A., Moritz C., Houghton V.M., Seth R., and Mayeron Me, 2003. Role of the corpus callosum in functional connectivity. American Journal of Neuroradiology 24, 2018-2212. PMC Free Article. Reichler Me, McLeod A.M., Snyder Oz, Powers W.J., Gusnard D.A., and Schulman G.L., 2001. A default mode of brain function. Proceedings of the National Academy of Sciences, USA 98, 676-682. PMC Free Article. Raj D., Paley D.P., Anderson A., Kennan R.P., and Gore J.C., 2000. A model for susceptibility artifacts from respiration in functional echoplanar magnetic resonance imaging. Physics in Medicine and Biology 45, 3809-3820, Rasmussen R., Nicholas E., Peterson N.C., Dietz A.G., Shu Q., Sun Q., and Nadergaard M., 2019. Cortex-wide changes in extracellular potassium ions. Parallel brain state transitions in awake-behaving mice. Cell Reports 28, 1182-1194. PMC Free Article. Reshubsky A., Wojtasiewicz T.J., Michael C.B., Bouchard M.B., Timmerman D., Youngerman B., McGovern R.A., Otten M.L., Cano P., McCann 2G.M., et al., 2013. Direct intraoperative observation of 0.1 Hz hemodynamic oscillations in awake human cortex. Implications for fMRI. Neuroimage 87, 323-331. PMC Free Article. Reimer J., Frauderakis E., Cadwell C.R., Yatsenko D., Denfield G.H., and Tolias Az, 2014. Pupil fluctuations track fast switching of cortical states during quiet wakefulness. Neuron 84, 355-362. PMC Free Article. Reynolds M.L., Gregory T.F., Blaumanis or Fujimoto K., and Grady P.A., 1985. Evidence for a paravascular fluid circulation in the mammalian central nervous system, provided by the rapid distribution of tracer protein throughout the brain from the subarachnoid space. Brain Research 326, 47-63. Revel A., Galit C., Orea V., Chapuis B., Beres C., and Julian C., 2012. Effect of chronic cervical ganglionectomy on the spontaneous variability of internal carotid blood flow in the conscious rat. Experimental Psychology 97, 564-571. Rosenegger D.G., Tran C.H., Wamsteker Cusselin G., and Gordon G.R., 2015. Tonic local brain blood flow control by astrocytes independent of phasic neurovascular coupling. Journal of Neuroscience 35, 13463-13474. PMC Free Article.